Have you ever purchased a tissue culture plant and in a few days or a few weeks it looks like this? Well, in this video I want to show you how you can still clean this plant to go like from this to this. So let's get started. How's it going, guys? Welcome back to another video. So today we are dealing with endogenous contamination. This is a contamination that is inside of the plant. So you buy a plant or you sterilize the plant yourself and you do everything correctly. You sterilize the outside and in a few days, a few weeks, then it starts growing bacteria that looks like this. Or you buy a plant and it arrives looking clean, but then in a few days it just turns like this. This is endogenous contamination. It's a bacteria or a fungi or yeast that is inside of the vascular tissue of the plant. So even if you re-sterilize it with bleach, then still the media turns on like this in a few days or in a few weeks because it's inside of the plant. So let me show you a couple examples. So this is a Philodendron Pluto, and it's severely contaminated. Now I went from this to this. These were basically the same level of contamination, and you can see this one is now clean. And I want to show you how to get it back to clean in a few moments. But first I want to show you some examples. So we have something like this. This is an orchid, and you can see this yellow bacteria that is growing here. So we also going to treat this plant. This Philodendron domesticum, this one has been here for a while. So this is from March. And over time it has leaked quite a bit of bacteria. You can see there on the bottom. So this is it should have been subcultured a long time ago. I want to take the best looking plants, I want to clean them, and I want to replace it back in tissue culture. I have some other examples here. So this is agave, and again I had this kind of yellow bacteria. You see this yellow bacteria. So I'm showing you different examples. This is vanilla orchids. You see the yellow bacteria. This is more vanilla, and then it has this bacteria also there. Yeah, all of this is vanilla, and they have they were very they have a lot of endogenous contamination, and I want to deal with that on this video. All right, so the method that I use and how I was able to get some clean plants from some severely contaminated to clean plants mostly relies on the media. So let's start by making the media. So fairly simple. It's pretty much the identical way on how you make the media. So let's start. I want to make a liter of media. So I will start with about 800 milliliters of the still water, and I will make I will use MSP09 media. This is your regular MS media, and I want to do full strength. So I need 4.43 grams of MS media. Okay, so 4.43 grams of MS media is sugar. This is just regular stable sugar from the grocery store, and I will add 30 grams of sugar. Right, so 30 grams of sugar. All right, next is the secret ingredient on cleaning contaminated plants. It's going to be PPM. This is plant preservative mixture. This is for a product that we develop here in Plant Cell Technology. This is used to prevent the growth of fungus and bacteria. And if used correctly, you can also prevent or even eliminate endogenous contamination. So let's add the PPM. Now, the typical dose of using a PPM is between 0.5 milliliters to 2 milliliters of media, or maybe all the way to 4. However, for endogenous contamination, when you have a contamination that looks like this, you can eliminate the contamination by adding more PPM. How much PPM? Between 5 to 10 milliliters per liter. So in this case, I'm making 1 liter of media, and I have a very severe contamination, as I showed you a moment ago. So I will add 10 milliliters of PPM. There we go. So 10 milliliters of PPM into my 1 liter of media. Next, I will add the rest of my water. Perfect. All right, so I have 1,000 milliliters of water, and I want to calibrate the pH. Now you notice that for this media, I'm not adding plant growth regulators. In this case, at this moment, I don't really care about adding hormones to my media. All I care is about cleaning them, so I can have them clean and then transfer it into some more regular media, multiplication or routine media. But let's calibrate the pH. So my pH is going to be very low. And I need to raise it to 5.6 to 5.8. All right, so I'm at 5.8, exactly what I want it to be. All right, so now that my media is ready, I will add my gelling agent, and I want to be using gelling gum. I want my media to be as clear as possible so I can see the contamination much better. So I will be using the gelling gum. And I don't want the media to be too hard, so I'll only be using 2 grams of gelling gum. So I have the 2 grams of gelling gum. All right, so I have sterilized my media. I have poured it into 5 racks of test tubes. So I have 105 test tubes. So yes, this is something that it is more expensive to do, as I'm adding 10 milliliters of media. So in total, you are probably spending close to 10, 11, $10 to $12 in media for 1 liter. Just on adding all the ingredients, especially for the PPM, that's roughly around 10 milliliters of PPM is roughly around $10, which I know is going to be expensive, but that's why we divide it into a lot of test tubes. So these test tubes are ready to go. You can see very clear gel, perfect. So we're going to be able to see it. You see, I only have 10 milliliters, which is enough for what we do. And we're only keeping the plants here, or we are cleaning the plants. So there is, we are not like keeping here for that long. Maybe just a couple of weeks for them to have the PPM to be able to get inside of the plant and then do its job, and then eliminating and inhibiting the growth of those or the fungus and bacteria, or especially the bacteria or yeast that is inside of the plants, and to kill all the contamination and have clean plants once again. So I have my racks of test tubes. So I want to bring the plants, the contaminated plants, and let's clean them and let's place them into the test tubes. All right, so let's get started with the nastiest plants. So these are the Philodendron Pluto. You can see they're all nasty. Oops. There we go. And I brought the microscope. I don't need the microscope. It's not needed for what I want to do, but this is just so you can see better what I'm doing, just so I can show you better. All right, so let me brought the microscope here. Let me grab get the plant out of the container. All of this has to be done under aseptic sterile conditions, even though like the plants are very contaminated. You still want to keep everything as clean as possible. So basically what I need to do is I grab with my forceps, with clean forceps, I grab the part where it's not like nasty contaminated bacteria, and with the other side I will remove all of this. If I just put this into put the plant into clean media, a lot of bacteria will grow on it. So I just want to remove all of them, or as much as possible. Let me switch sides. I'm sorry, the image is very dark. Let me see if I can adjust the image. I'm going to peel these larger outer leaves as well. So all of this base I don't want. So I want to cut that. I want to expose fresh plant tissue. So this is what I want. So I remove all of the contaminated part of the plants, and this is what I want. You can still see a little bit of bacteria hanging out there, but that is what the PPM will essentially take care of. So I want to grab a sterile test tube. And put it in the plant. All right, so I need to make sure I have a good contact with the media. So go as deep as you can. So you see, just like that. And now I have to repeat with the rest of the plants. Obviously, my plate is very contaminated. So you need to, after every single plant, you need to make sure to do a good cleanup. Re-sterilize your surface. All right, here is the second plant. So once again, pull my plant out of the contaminated media. Very nasty contamination. So I don't want anything from here. Get it out. And all of the bottom leaves. There we go. So that should be good. And now I will place it in a test tube. And that's it. That's the second plant. Right, for my next example, I have these orchids. You can see it has contamination all around. So I have multiple plants. I'm going to start with the larger plants first. All right, so here is the plant. And with this one, I have to be very, very careful not to damage the grow point. So all I'm going to do is just, with my scalpel, remove all of this kind of the dead part of the plants, this part, and I will try to remove this bottom leaf very, very, very carefully. Just peel it up like that. And I think that's all I want to do. So that's all I'm going to do. And now I'm going to take my test tube and place it into the test tube. There it is. All right, so dead leaf will have to come out. So make sure I have a good grip with the plant. That can come out, and all of these I can trim that up as well. This bottom leaf I want to also take it out. Okay, perfect. And that's all I'm doing with this plant. Take my test tube and get the plant in there. So you see, very simple. It's just basically removing all the bottom leaves to make sure that to start with a clean part of the plant, and that's it pretty much. Then obviously in between each plant you have to re-sterilize your surface area. You're working with, like, let's say I'm working right now with a glass cutting board, but if you're using plates, just replace plate, or paper towels, just replace the paper towels every time. But yeah, make sure to give it a good sterilization after every single plant. So yeah, you will be using a lot of paper towels. Again, for the paper towels, it's only one use, so goes to the trash. All right, we have another example here. This is a vanilla orchid, and these are relatively easier to clean. We have the plants, and they can be fairly easy remove the contamination. As you can see, we have the plant. The contaminated part is right here at the bottom. So what I'm going to do is just cut just on top of that, and that's the part that I want to place into my new tube. So let's do that. All right, so I have the plant, and you see all of this as bacteria right there. So I don't want any of that. So I can cut that one there. I also want to cut this one here, so I want to keep these two parts. I'll probably even cut this part. I mean, if I want, I can start multiplying this as well, so I can just also absorb culture them. So I can just also cut that one there, and this one I can also split it if I wanted to. But now I take my plant, goes in the medium right there. This is a different plant. This is agave. Has more of this yellow contamination. Oh wow! More of the yellow contamination that I need to get rid. Right. So for this plant, as I was taking it out, basically I ended up dividing into three parts. The top part came out by its own, so that one is clean. So what I'm going to do is I'm going to straight up place it into a test tube. I'm not even going to cut it. I don't need to, because it's not contaminated. So that can go into the test tube. Same thing with the second piece. It's also clean. Doesn't have any of the bacteria. So it can also go into the test tube. Let's see how the third one looks like. Let's see if there is a lot of bacteria, or if I can use. There is not a lot. There is not a lot. I mean, obviously they have bacteria on it. I probably just going to let the PPM do its job in this case, because obviously trying to clean this up is going to be super difficult. So I'm just going to let PPM do its job. Maybe you can remove as much as the old media, because obviously it's going to be contaminated. So I'm just going to do that. If you can probably also rinse it with the sterile water to remove all of this old media. There we go. So I want to place this part in a clean test tube. Next plant is this very old Philodendron domesticum. Should have been subcultured a long time ago. It's covered with a bunch of bacteria at the bottom. So let's get started. Let's clean it up with some alcohol. Clean my hands. Let's take out. Well, I guess this will have to come out as one thing. But what I'm going to do is remove the individual plants first. So remove the individual plants, place it to the side. Don't want any of this nastiness here, so just get it out. I think that's all I'm keeping. It's much better. Just take all of this out, cut the base. And that's good. Good cleanup. Now I can go into the test tube. All right, and the last plant and the most difficult by far is an alocasia. This alocasia, again, I know many people have issues with alocasias, getting with endogenous contamination. I have a lot of issues as well. This is probably the hardest one to get it clean. So let's give it a try. So let's take it out. It is very, very dirty. I already tried to clean this before, and as you can see, it got contaminated once again. So the most difficult plant to get it clean. So you can see all of that nasty bacteria there. Well, you cannot see it. Now you can see it. You can see all of the nasty bacteria that is in the on this plant. So it's almost a goner, but I'll try to do my best to see if I can clean it up a little bit better. You know what I'm going to do on this one, because it's so nasty, I will use sterile water to rinse it. Oh yeah, so all right, just rinsing the explant. First I just sterile water to try to remove all of the nastiness. So I already washed it a couple times. So you have all the nasty bacteria there. So I'm just doing that. I just rinsing with the sterile water until my water looks clean. All right, I think that's going to be good enough. So what I'm going to do next is cut the plant. So let's go back under the microscope. Let's take my plant out. Take the scalpel and let's cut it. So all of this dead plant tissue I do not want. So I'm going to position my plant straight down like this, and I'm going to slice it to expose green tissue. Just like this. All the way around. I don't want to see any dark plant tissue. Can also cut the end. And here can make it smaller. There we go. So that's my meristem right there. So that's what I'm going to place in my tube. So you can see now it looks much more cleaner. And hopefully the PPM will take care of the rest of the bacteria or the endogenous contamination that is inside the plant. So I'm going to take a test tube, take my plant, and it goes in the media, very deep in the media. Just like that. So there is my allocation. So you see I cut quite a bit around. Everything that it was nasty, everything that is that looks dead. You can see all the everything that I removed, all of this. So with that, hopefully by now, I have a clean plant and the PPM will take care of the rest of keeping it clean. All right, so it has been a week since I cleaned the plants and I placed it in media with a high amount of ppm, 10 milliliters per liter. And the first one that I want to show you here, these are the ones that I did not clean. So these are, I didn't fully clean all of them. I wanted to have like a comparison on how these ones are looking, the ones that I didn't clean and the ones that I cleaned. So let me show you, like for example, agave. This is how the agave looks like. The ones that I did not clean, right here. And where is my agave? Let's see, where is my agave? Here is my agave. So this is the agave that I cleaned. You can see it looks much cleaner. I don't see contamination on them. Let me see the other one. So here's the second one. No contamination. Maybe just barely, slightly, a tiny bit. But very good. And this is the third one. And this one, I do not see contamination on this one. Great. So that's great. For the agave, it works pretty well. This is the vanilla, orchid vanilla orchid. You see it has the contamination. A little bit on the sides, like this one, it has the contamination just right there. So these are the ones that I did not clean. And they have a little bit of contamination right there. So the ones that I clean is basically this whole rack, the entire rack. And I can tell you that they all look very clean. They all look very clean. Like I say, it has been a week, so today is the 19th of November, and they were done on the 13th. And they look very clean. Something I forgot to mention is that when I place the plants into the media with high dose of ppm, it is normal that in the first 24 to 72 hours, you see some contamination start coming out. But then after a couple more days, you actually see that the contamination starts to disappear. So that is the ppm doing its job. But it takes a couple of days. So it is normal, perfectly normal, that you see contamination showing up in the first few days. And then ppm usually takes care of it. And by 1 week, it usually is very clear results where you can see if it works or if it doesn't. And then over the next period, the next few days, 2 to 3 weeks, it stays relatively clean. So like these other ones, they've been here since October, and they are very, very clean, and they are ready to go into use regular media with regular dose of PPM. So very happy with that. This one has a tiny, tiny, tiny bit of contamination. Again, it may take a second round. Like I may have to, if it still has a little bit of contamination, you may have to subculture once again into like the same media with like the high dose of PPM. But if they are clean, then they can go straight. They can go back into the media with a less amount of ppm, so 1 to 2 milliliters of ppm. So yeah, this one has a tiny little bit, but not much. It's just very little. This one has a bit more, but this probably has a lot more contamination. You can probably see it right there, right at the edge, right there. So this one will need a second run of ppm for sure. This one probably as well. But I mean, the overall, very good. Now let me show you like the ones that I clean, like they had the most contamination were the Philodendron Pluto, and let me show you how they look right now. So here these are from the one from class. So this is the one from the 13. So you remember how contaminated they were? Look at this. It has a little bit. You can see a little bit of like a dome, like a dark dome down on the bottom. So a tiny little bit contamination, but it's not bad. A little bit of endogenous, but it's not bad at all. This one as well, right there. You can see a little bit of a darker shade right here at the base, but very, very clean otherwise. Like they look very clean. And the spirusanti, that they were also very contaminated. Check it out. Much cleaner. Now the most important part about like is that it's clean, but it's also healthy. The plant, it looks fine. The plant looks fine. They have tolerated the 10 milliliters per liter of ppm just fine. And yeah, just fine. And then they will, as soon as I place it back into the, obviously they are probably not going to grow that much in this media, as it doesn't even have any hormones. It's just the MS media. But they have done very well. Now the orchids, these are the orchids. You remember they had this yellow bacteria. Very clean. Very clean. These orchids are very notorious for like endogenous contamination. Very clean. This one, the test tube is a little dirty, so it's no bacteria, but the test tube is a little dirty on the bottom. But yeah, otherwise, very, very clean. So I'm very happy with the results. And again, kind of the biggest thing is like, all right, so I made 5 racks of test tubes. Obviously, I only have 3 because I didn't need the other ones. The other ones are still there. I didn't have that many plants. Well, I can use it for the rest of these contaminated plants. But I did 5 racks of test tubes, 105 test tubes. Yes, I used 10 milliliters of ppm, which it costs roughly, if you use the smaller bottles, it costs roughly $10 of ppm. And then obviously with the media, so we are talking roughly around $12, $11, $12 of the media that is going to cost you on the media. But you're making, like for example, I'm making 105 test tubes. So if I divide that into the 12 into the 105 test tubes, I mean, we're talking somewhere between like, what, 9, 10 cents per test tube. So if you think 9 or like 10 cents per test tube is worth potentially saving a plant that costs a couple of $dozen or maybe even potentially hundreds of dollars, I think that is worth it. The only thing that I still have a challenge, where is it? Here it is then the allocation. So the allocations are very, very notorious, very, very difficult to clean. So this one, I can see is growing now, but it's also growing contamination. You can see it's still is contaminated. So I had to try to clean it once again. But these are the ones that are the most difficult to clean. So I need to try to figure out if I can come up with another protocol to clean allocations because they are so difficult to clean, even like with the high dose of PPM. So I'll try to figure out something else, and I will let you guys know. But overall, for the rest of the plants, the PPM has done very fantastic. It has eliminated most of the, I would say like 90+% of the endogenous contamination. Some of the plants may have to go to a second round of a PPM, but for most of them, they can go back into the regular media. And you see, it only took like a week. You can probably leave it like a week. Usually, I recommend like a week, maybe 2 weeks, and then go back to the regular media. As obviously with the high dose of PPM, it will stunt the growth of the plants a little bit, so or maybe slow down the plants or the growth of the plants a little bit. You can see it doesn't actually damage as much or like doesn't kill the plants. 10 milliliters of ppm, the plants that still look very healthy, look fine. But the plants, they would definitely would not grow as quickly as the plants growing in regular media. So only a couple of weeks in this media, and then they can go back into the regular tissue culture media. So I think that was everything for today. Again, stay updated. So I will try to find another protocol for allocations. But other than that, thanks for watching. I will see you on the next one, and
Thanks
goodbye.
for watching the video. If you are enjoying these tissue culture videos, make sure to follow us on social media for more informative content. And if you are interested on conducting your own tissue culture experiments, make sure to check us out at plantcelltechnology.com for all the products you need to get started. Use code FP10 for 10% off on your first purchase.